Okay, good to see you again. These are our notes for Tuesday, November 4th. In your concept map, in your book, or in your packet, I'm sorry, you'll have a couple pieces of information. We'll title these together, so make sure you're paying attention. The title of this concept map should involve Life is Cellular, so write that down at the beginning at the top of your page. Life is Cellular. Excuse me, for your main topic in this first concept map number 1, write Life is Cellular in that main bubble as well. Okay, so what we're going to do is we're going to go through this information, and I have it mapped out in A, B, C, and D, so those are the bubbles that we're talking about. So we know life is cellular. Some of the key points. We know, and it's highlighted here, and I've also lettered it, there's a common structure that makes up every living thing, and that is a cell. So bubble A, right there is a common structure that makes up every living thing, and that is a cell. For bubble B, we also saw this in our Eukaryopolis video. In 1665, Englishman Robert Hooke used an early compound microscope to look at a thin slice of cork of a plant. Cork is a plant, so this was the first experience folks had at looking at cells. So bubble B, 1665, Englishman Robert Hooke used an early compound microscope to look at a slice of cork. For component C, part C, in our C bubble, we know that this tiny piece of cork through that microscope had tiny chambers, and that's what Hooke classified as being cells, empty cells. So for bubble B, we know the tiny empty chamber was characterized as being a cell, and that's how cells got their living name, got the name associated with them. We know that for part D, today we know that cells are not empty chambers, but contain living matter. So part D, cells contain living matter. Okay, continuing on for our second concept map, we'll title this second map the cell theory. And as we talked about, this was actually one of our SLL assessment questions. The cell theory kind of defines what we're talking about, the characteristics associated with cells. And we'll use this concept map to do that. So in your main bubble, concept map number 2, type in cell theory, please. Alright, and we know that the cell theory, as we made mention, defines cells. It defines what cells describe, components associated with them. So for bubble A, we know that cells are a basic unit of life. And we know two things. This will go in with bubble A as well. All plants and animals are made up of cells. For bubble B, we know that cells are produced only from the division of existing cells. So cells come from other cells. And what we'll do with bubble C, D, we'll actually have to add a bubble E, but the last three components of this topic identify the cell theory. And the cell theory states, so for C, D, and E, write each one of these components. So we know all living things are composed of cells. We know the cell theory also states that cells are the basic unit of structures and functions in living things. In our third component, which we'll have to add, bubble E, we know new cells are produced from existing cells. For our third concept map, we're going to explore the topic of the history of cells, and we're going to use this concept map to break down a timeline that identified the evolution of understanding more about cells. So for this, we're actually going to have to add 2 additional bubbles, so we'll have A, B, C, D, E, and F. And once again, the title of this concept map involves the history of cells. So in addition to our concept map, as I said, we're going to use that as a tool to break down this timeline. What we're looking for in this timeline basically deal with the date and just a brief summary of what happened during that date. So we know the first one we're talking about is 1665. Robert Hooke identifies sections of a cork that are empty. He calls those cells. 1674, Anton van Leeuwenhoek. He observes tiny living organisms in drops of pond water through a simple microscope. And continuing on in bubble C, we know in 1838 Matthias Schleiden concludes that all plants are made up of cells. And in 1839 Theodor Schwann concludes that all animals are made up of cells. In addition to this, in 1855, Rudolf Virchow proposes that all cells come from existing cells. And this, in essence, completes the cell theory that we previously talked about. And in conclusion, in bubble F, we know in 1970, Lynn Margulis, she proposes the idea that organelles, which are tiny structures found in cells, were once free living cells themselves. And continuing on for prokaryotic and eukaryotic cells, we talked about this in relation to one of our additional learning goals. For concept map number 4 in our main bubble, we're going to introduce prokaryotic and eukaryotic cells. So if you could, please put that in the middle bubble, that concept map, and we'll go from there. Okay, and looking at prokaryotes and eukaryotes, we know certain differences and similarities exist. For concept A in our concept map, we know that despite their differences, all cells have two characteristics in common. So what we need to write for bubble A is we know that all cells are surrounded by a barrier called a cell membrane. And for bubble B, we know that these cells also contain molecules that carry biological information. So we know some similarities exist between prokaryotes and eukaryotes. We know bubble A, all cells have two characteristics in common. The first being they're surrounded by a barrier called a cell membrane. Bubble B, the second similarity, we know that cells contain molecules that carry biological information such as DNA. We also know that certain differences exist. We know depending on what type of category of cell we're talking about, often determines whether or not they have a nucleus or not. But to start, we know that bubble C, we're going to talk about a nucleus. So the nucleus is a large membrane enclosed structure that contains the cell's genetic material. We know that as DNA. Okay, so what types of cells have a nucleus and what type of cells don't have a nucleus? So for bubble D, we're going to identify this difference. So we know that eukaryotes are cells that contain a nuclei, while prokaryotes are cells that do not contain nuclei. Okay, next concept map. We're going to start concept map number 5, and our main concept we're going to talk about involves prokaryotes. As we mentioned, prokaryotes are kind of unique. The things that define them make them unique compared to prokaryotes. Eukaryotes. So prokaryotic cells are generally smaller and simpler than eukaryotic cells. So for bubble A, we know that prokaryotes are generally smaller and simpler compared to eukaryotic cells. We know that prokaryotic cells for bubble B have genetic material that is not contained in the nucleus. So we know that prokaryotes are cells that do not contain nuclei, right, a nucleus, but they also have genetic information. But prokaryotic cells have genetic material that is not contained in the nucleus. It's contained in other parts of the cell. For bubble C, we know that prokaryotic cells grow, reproduce, and respond to their environment. And for bubble D, we know that prokaryotes, an example would be bacteria. And finally, concept map number 6, our main bubble of this map, deals with eukaryotes. We've already spoke to a little bit about what makes prokaryotes unique, and the second component here will finalize with eukaryotes and eukaryotic cells. Alright, in conclusion with our last concept map, we know, once again, we're talking about eukaryotes, and for bubble A, we know eukaryotic cells are generally larger and more complex than prokaryotic cells. For bubble B, we know eukaryotic cells generally contain dozens of structures and internal membranes. This is something we'll talk about later. We know these are referred to as organelles. We'll talk about that the next section. For bubble C, we know eukaryotic cells contain a nucleus in which their genetic material is separated from the rest of the cell. Once again, this is a difference compared to prokaryotes, whose genetic information is found in something other than the nucleus. And finally, bubble D, we know eukaryotes are generally large multicellular organisms. Examples would include plants, animals, fungi, and protists. We will cover this topic briefly tomorrow. If you have any questions for class, let me know. Once again, if you have any suggestions as well, keep me posted. Good luck.